This is supposed to be the first joint degree program between the university at Pajajaran, so it's called UNPAD, and Rutgers, between the graduate schools there. And Alvin is the first student, and I'm happy to observe that Alvin has done quite well under your tutelage. And I'll let you say about him, because objectively I cannot comment on that. All I know is Alvin is a very enthusiastic young individual.
Yes, so we're really proud of Alvin. So he is, this is a double degree program as far as I understand.
Right.
He's going to get a MS in Clinical and Translational Science at Rutgers. His grade point average is 4.0. That's perfect.
That's on the Richter scale, right?
He's also getting a master's in pharmacology at the University of Pajajaran. I don't know if I pronounced that right.
Yeah.
And I don't know why his grade point average is only 3.94. What happened?
Oh, what a slacker. What a slacker.
His advisors there, who are all with us, are Dr. Kerry Listari and Dr. Harry Hammond. Did I miss somebody? Right? Okay, Alvin. Alvin is a MD who has had a year of internship before starting his PhD, the MA program. He has been one of my best students among many really good students. And he's worked hard. He's accomplished quite a bit. And his master's is not quite a PhD thesis, but it's getting there. We're really proud of him. And I could say a lot of nice things about him, but I think you all know him and know that as an example of a joint program and a student that's been in this joint program, debut, you picked the first one was really great. Anybody else want to say anything about Alvin? Emily, you know him not well though. Right.
Well, I met him at the New Year's party with my daughter, and we were both very impressed, and we were so worried about her, about COVID, whether he would be lonely all by himself. So I'm so happy that he did wonderfully. So I'm very happy to meet him and to see this day come.
Okay, so thank you, Debu, for giving us Alvin. And without further ado, as they say, Alvin, take over.
So, So, good morning, everyone. Very good morning and maybe good evening, very good night in Indonesia. Thank you for joining us in my public defense. And this is a very, a very honored moment that we have been waiting for more than a year, I guess, during this pandemic season. But actually, we made it. Before I start, I would like to introduce all my committee members and the thesis examiner from both universities. I will start from my primary advisor, Dr. Joseph Albertino from the Cancer Institute of New Jersey. Thank you for guiding me all through all these times. And then I would also like to introduce you to Dr. Kerry Listari. He's a professor of pharmacology at Universitas Pajajaran. I would like also to introduce you to Dr. Harry Herman. Dr. Harry Herman is an orthopedic surgeon and also associate professor of orthopedics in Universitas Pajajaran. Then the thesis examiners, I would like to introduce the chief of the committee, Dr. Sharon Pine from the Cancer Institute of New Jersey. And then I would also like to introduce Dr. Debabrata Banerjee. As my former program director, Dr. Banerjee, one who recruited me to study at Rutgers. And then I would like also to invite Professor Hanggono Ahmad. He was the former provost of our university and the one who recommended me to study here. Then we also have Professor Anas Subarnas, Professor of Pharmacology at Universitas Padjadjaran. Last but not least, we have Professor Rizky Abdullah, the recently inaugurated professor at Universitas Padjadjaran. So welcome for all committee members, and I would like to ask the permission for sharing my screen for my presentation today. All right. Can you all see my screen?
Yeah.
All right. So again, very good morning and good evening, everyone. Would like to present my work for the past year about synergistic antitumor effect of a NAMT inhibitor and an AD kinase inhibitor in mutant IDH1 cancers. Last year, when I came to Dr. Bertino's laboratory, I found this paper published in the AACR journals, and I see that some people has been working very hard on this paper. I remember there was Phil, there were Emily, and also Debu also working on that. So I come to Dr. Bertino and ask, can I think we can improve your study by choosing the right candidate for the treatment? And then Dr. Bertino challenged me to do more. So here it is. We did some introduction, and we will have some introduction and some review there about what is the NAD, about the cancer, and also the biosynthesis pathway of NAD, and how the NAMT enzyme and NAD kinase enzyme play the role in working on the cancers and in treating cancer. And then I will share the result of our study for the past year. So as an introduction, we know that there is a molecule, it is a very famous molecule for scientists called the NAD, or the nicotinamide adenine dinucleotide. It's a coenzyme that mediates the redox reaction in all living cells, even if, whether it's human, animals, or plants. And it plays a key role in energy generating metabolic reactions in the cells. NAD helps the cells to maintain the cell cycle, the transcriptional regulations, to repair DNA, cell signaling, and also as an anti-apoptotic regulation. The cancer cells have a unique energy metabolism for sustaining rapid proliferation. We know in around 1950, Otto Warburg discovered that aerobic glycolysis prevert the normal respiratory chain in cancer cells, right? And then the amount of energy from the aerobic glycolysis is actually less than the energy produced by the normal respiratory chain. And that's why how cancer cells actually produce significant amount of energy to support the cancer survival in a short period of time. And then, cancer requires continuous and elevated presence of NAD in both glycolysis and gluconeogenesis. And that's why if we get or reduce NAD, it will be a disaster for cancer cells. So there was a literature out there saying that early on, NAD levels, NAD will give a protective effect against oncogenesis in the very early process. But later on, as the disease advances, NAD makes the cancer survive or worsen the disease. If we take a look at the NAD biosynthetic pathway, NAD is generated through three biosynthetic pathways named the nicotinic acid pathway, the NOVO pathway, and the NAD salvage pathway. what we are going to learn today, what we are going to discuss today is about the NAMT enzyme, the NAD sulfate pathway, and the NAD kinase enzyme that convert NAD into NADP and further NADPH. So this is the model that I have been generated through these years with Dr. Bertino and also Gulam in the lab. We learned that in the carboxylate acid cycle, we know that as a Krebs cycle, the molecule alpha-ketoglutarate is converted by the mutant IDH or isocitrate dehydrogenase enzyme into B2-hydroxyglutarate, which non-metabolite which promotes oncogenesis by inhibiting the dioxygenases. And then this oncometabolite will promote NAPRT hypermethylations that inhibit the NAPRT enzyme in the nicotinic acid pathway. If the pathway is inhibited, then the amount of NAD generated, the generated NAD will be decreased. And that's how we are going to take advantage of the NAD sulfate pathway, because 90% of NAD metabolism, NAD production comes from the NAD sulfate pathway. So again, about the NAMT enzyme that becomes the star of our study this year. NAMT enzyme is actually an enzyme that plays a key role in maintaining the NAD pool. Okay. The increased expression of NAMT has been linked with a lot of diseases, such as obesity, non-alcoholic fatty liver diseases, diabetes mellitus, and also, in particular, is cancers. Increased NAD pool, which is actually associated with chemotherapeutic resistance. And there's a study there when that knocked down the NAMT. It shows that cancer cells are more sensitized to increased reactive oxygen species and cell death. And we did some literature research about how mutant IDH cancer cells are mostly sensitive to the NAMT inhibitors in vitro. So from those literature, we saw that we can see that the mutant IDH cancers actually showing depleted or decreased NAPRT expression, meaning that less NAD are developed. Less NAD developed and showing that the cells are highly sensitive to NAMT inhibitors. And the NAMT inhibitors also show work synergistically with other chemotherapeutic agents in vitro. We can see that most of the cocktails, the treatment, cause decreased cell viability, decreased NAD levels, as well as increased apoptosis and cell death. The NAMT inhibitors also work synergistically with other chemotherapeutic agents in the animal model. So for all this time, we can see that NAMT inhibitors can be combined with the other treatment to decrease tumor volume with minimum toxicity. Again, the concept of drug discovery and development. When you start working from the in vitro and the clinical study, when you succeed with the clinical study, we have to go through the clinical trial. The problem starts here. There was a very old, I think not too old, study in the Europe starting at 1999, withdrawn in 2012 about NAMT inhibitor. The NAMT inhibitor recommended dose. The recommended doses was reached, but not recommended for further trials because of disease progression and severe adverse events experienced by the study subjects. So the NAMT inhibitors has a problem with lack of efficacy, with the efficacy issue when it goes into trials. The phase 2 study about NAMT inhibitor was terminated. It's because of, again, lack of efficacy and also adverse event. So it's both. It's actually just both efficacy and safety issue. So what we learn here? Some people start to postulate it. Some people postulated that it might be because of the extracellular nicotinamide that caused failure of the trial. Right. I'm showing you this again. Then remember, we start with the alpha ketoglutarate that is converted by the mutant isocitrate dehydrogenase to produce D2-hydroxyglutarate. D2-hydroxyglutarate is the oncometabolite that promote oncogenesis and inhibit the NAPRT enzyme, leaving the cells to solely depend on NAD salvage pathway to create or to produce the NAD in the cells. That's why we call NAMT as the rate-limiting enzyme in NAD generation. So the D2-hydroxyglutarate accumulation and the NAPRT hypermutation, the mutant IDH1 are potential candidates for NAMT inhibitor trials due to lack of the nicotinic acid pathway. And then until this time, there are no NAMT inhibitor trials conducted in mutant IDH1 or 2 cancers. Talking about NAD kinase as the enzyme that convert NAD to NADP, NAD phosphate. So NAD kinase is converting NAD kinase converts NAD into NADP. That will be further converted into NADPH. The problem starts when NADPH is highly accumulated. It can be a reductive biosynthesis and can provide defense against reactive oxygen species in cancer cells, meaning that high NADPH level will make the cancer cells survive. A little bit profile of the properties of NAD kinase was firstly discovered by Kornberg in Germany around 1950. It was observed in algae, in high-level plants, and also animals. But then until maybe 50 years later, people start to discover NAD kinase in human and found that NAD kinase is highly expressed in human placenta, in liver, and also in the colon. I know that this is a very old Northern blot picture, but this is what we can find in the literature before. And this NAD kinase, it's actually showing some properties that is really nice to learn. And then again, when we learn about human NAD kinase and cancer, the interdependency between these two entities, I learned it from Emily and Yiching. Thank you, Emily. So the role in cancer treatment was not fully revealed until 2012. That is when Emily started to learn and to research about this. The thionicotinamide or NADPS, and is an NAD kinase inhibitor that degraded the hydrofolate reductase and inhibited cancer growth. So yeah, again, this is a study from Yiching and Emily back at the time. We can see that NADS and NADPS exhibit some cytotoxicity profile in cancers, but also showing safety profile also. Again, when it was combined with other drug, it shows that NADPS works synergistically with methotrexate, and also Phil's did the study in 2015 showing that thionicotinamide, a prodrug of NADPS, also exhibit efficacy in treating cancer cells. And it's also have a synergistic activity with irinotecan to the C85 cells in vitro, and also in vivo study. So talking about research significance and novelty, NADPS, Yiching state that NADPS enhanced VHFR degradation in 2012, while Fields stated that NADK inhibitor lowered NADPH pools, inhibited cancer cell growth, and worked synergistically with other chemotherapeutic agents. And there's a study in 2016 published in JAMA saying that NAMT inhibitors failed clinical trials. So what we can learn from these 3 studies, that we have to choose the right cancer candidate to be treated with the right drug or the right compound that we have. And then why don't we, at the time I was thinking and say to Dr. Bertino, why don't we try to combine the inhibitors that we have? We have the data from NAD kinase inhibitor in 2015. Why don't we just combine it with the NAMT inhibitors, a well-known NAMT inhibitors and then try to treat it in the mutant IDH1 cancers, I hope we can take advantage from this combination study. So based on that discussions, I told Dr. Bertino at the time and say that, why don't we establish the synergistic effect of NAMT inhibitor and NAD kinase inhibitor in cancers, in the mutant IDH1 cancers actually, with the hypothesis breakdown coming that, anakinramide has successfully showed cell death and decreased metabolic product synthesis in C85 colorectal cancer cell line. IDH1 mutation generates the D2 hydroxyglutarate, inhibits nicotinic acid pathway in NAD synthesis. Therefore, we hypothesize that both inhibitors may cause reduction of NAD that may be sufficient to sensitize mutant IDH1 cancers. So we learned, so at the time, we have some colorectal cancer cells in the lab. Based on my literature review, I found that HCT116, CT8, HT1080 were known or highly recognized as a mutant IDH1 cancers. And then we have this cell line, special cell line, C85 colorectal cancer cell line, isolated by Dr. Bertino like 20 years ago, and we still have it in the lab. So what we did at the time, I tried to, we made sure that these cells are the mutant IDH1 cancers. So we did the Western blot analysis, check for both IDH1 wild type protein and also the IDH1 R132H antibody. And then we discovered that, we learned that the HCT116, HCT8, and HT1080 cells express the mutant IDH1 protein. But C85 showing less expression of the protein. But we need to make sure that whether this cell line is a wild type or a mutant. What we did the PCR. We isolated the RNA and developed the cDNA, and then we sequenced it, and it was found that the C85 cell lines is actually a mutant IDH1 with the substitution protein it's a mutant R132E mutant. And according to that result, we did the cytotoxicity study profile, and we learned here that STF NAMT inhibitor STF 118804, our compound, shows cytotoxicity profile on mutant IDH1 cancers, ranging from around 150 nanomolar to around 70 micromolar in the cells. We also learned that nicotinamide, the NAD kinase inhibitor, showed cytotoxicity profile on mutant IDH1 cancers, and it reached the inhibitory concentration, or the IC50, around 2.5 up to 70 micromolar. As we had this result, we continued the experiment by treating cells with either single or combinations of the drugs in a fixed ratio based upon their IC50 using Cho-Telalai method. And after 72 hours of incubation, media was removed and cell viability was measured. This study, both synergistic activity was achieved in the HCT116 group with the combination, is the HCT116 data with the combination index value around less than 1. So it's actually a highly synergistic activity. Synergistic effect on HCT8 cancer cells were also shown with CI value around 0.53 up to 0.66. And we also have that C85 cell lines, which is a we have the CADF cell lines, which is a R132P mutant colorectal cancers that showed synergistic activity, but as we increase the dose, the trends are shifting to additive activity. And last but not least, the combination study on HT1080 chondrosarcoma cell line showed that we also reached the highly synergistic activity at lower dose, at the IC50 dose. As we increase the dose, the treatment start to show antagonistic activity at higher dose. Again, what might be this? Remember, it has been postulated that extracellular nicotinamide might be the cause of failure of the NAMT inhibitor clinical trials, and the majority of our experiments use RPMI media containing 8 micromolar nicotinamide. So I question myself, why don't if we what if we try to add more nicotinamide in the media, because it's because we also have like DMEM media that contains 32.4 micromolar nicotinamide just to grow the chondrosarcoma cells. Why don't we add some extracellular nicotinamide to the RPMA media until we reach the same concentration in the DMEM media? So we try that, and we can see here that we treat the HCT116 colorectal cancer cells with either single or combinations of the drugs according to the IC50. And our results indicated that HCT116 colorectal cancer cells need higher doses of each drug in the combinations to achieve the desired synergism in the presence of high level of nicotinamide. We can see from this figure that in the beginning, for IC50 for NAMT inhibitor and NAD kinase inhibitor in HCT116 were 35 and 25 micromolar. But as we increase the nicotinamide concentrations in the media, we can see that the IC50 of NAMT inhibitor became 105 micromolar, and we didn't even get the IC50 for high nicotinamide even after we increased the dose for 4 times. Right. So we further continues and question. So what happens with the metabolite when we try to when we apply the drug there? Starting or referring, let me introduce you from a very well-known feature that has been introduced since high school. The citric acid cycle. We knew that the molecule that we are studying is actually there, isocitrate and alpha ketoglutarate. You can see at the red box there, and the alpha ketoglutarate will be converted by the mutant IDH to produce 2-hydroxyglutarate that will inhibit the NAPRT enzyme. When NAPRT is inhibited, meaning that NAD productions is decreased because the cells only depend on the NAD salvage pathway, and then that's what makes our study. So we hypothesize in this study that the combination of STF anti-inhibitor and thionicotinamide NAD kinase inhibitor reduce the NAD and ADPH pool, as well as reducing the other important metabolites in the Krebs cycle, including the isocitrate, alpha ketoglutarate, and also end product of every bioenergetic cycles, adenosine triphosphate. So we plated 300,000 cells per well in the 6-well, supplemented with the respective media, and we treated them with the single or combination of the drugs according to their IC50, and leave it for 72 hours in the incubator. Metabolites were extracted, and we measured the metabolites using liquid chromatography mass spectrometry, and the result is showed that combination of both STF and thionicotinamide significantly reduced all metabolites compared to the control group. So if you take a look at the graph here, the figure here, the first figure is for the alpha-ketoglutarate. We can see that in the C85 cell lines, the first bar is corresponding to the C85, and the bar in the middle is the HCT116, and the third bar is the HCT8 cells. You can see that combination of both drugs showed you can take a look at the red bar and the green bar, showed significant reduce of alpha-ketoglutarate and also isocitrate in all treatment groups. We can also see that the same thing also happened for the hydroxyglutarate. We can see that statistically significant result of that decrease of the D2-hydroxyglutarate for both the combination group in every cells, in every cell lines, in all cell lines. Same thing also happened for the NAD, reduced NAD and the NADP profile. And then the last but not least, it's also showing that the NADPH, both NADPH and adenosine triphosphate also decreased significantly in all cell lines. We further continue to think that what if we try to knock down the IDH1 mutant? We hypothesize that by creating a wild type IDH1 knockdown cell line in a mutant IDH cancers might decrease the conversion from IDH alpha-ketoglutarate. Less alpha-ketoglutarate, again back to this picture, less alpha-ketoglutarate will result in less D2-hydroxyglutarate production. Less D2-hydroxyglutarate, by the same rationale, less D2-hydroxyglutarate will also have the NAPRT enzyme to be less inhibited. So try to imagine if we can knock down that IDH1 gene in the mutant IDH1 cancer cells. Less NAPRT will be less inhibited, NAD will be produced more, will be generated more, and the more NAD, the more NAD will be, there will be more NADPH. Therefore, we can have the cancer cells be more resistant to the treatment. And our results showed that we successfully knock down the cells, the C85 and the HCT116, and I can show you that cells are more resistant to the treatment. So if you take a look at the gray line here and the yellow and the blue line, we can see that the C85 cell lines is actually more resistant to thionicotinamide after the knockdown, and the HCT116 also showing the resistance to thionicotinamide. So for that discussion here is that it's important to choose the right type of cancer. So the idea of this is actually to choose the right candidate, choose the right cancer candidate for the treatment. And then we also confirm that our results are in line with the previous studies investigating the STF118804 in vitro and in vivo. We also confirm that the extracellular nicotinamide rescued mutant IDH1 cancer cells from dying. Then our combination study showed that the combination of both drugs achieved appreciable synergistic activity proven by the combination index value calculated by Cho-Telalai method. And our metabolomic data showed that inhibition of NAMT and NAD kinase decreases production of NAD, NADP, and NADPH. This might also decrease the D2-hydroxyglutarate production. The production of D2-hydroxyglutarate requires NADPH, not only the mutant IDH protein, but also NADPH. So here we highlighted the importance of NAD and NADPH pool in cancer survival. And by inhibiting or reducing the pools, the NAD and NADPH pools, we can disturb the bioenergetic cycle in mutant IDH1 cancers using both NAMT inhibitor and NAD kinase inhibitor, and that might be a novel strategy to treat cancers. I would like to say thank you to our committee in Universitas Padjadjaran and as well as in Rutgers University. I would like to thank Dr. Bertino. I know you say this every time, but I'm very honored to be the only students coming from Indonesia, and I might be the last student of Dr. Bertino. So thank you, Dr. Bertino, for having me as your student, to be the student of the word, father of chemotherapy. And also I thank Gulam, the postdoc in our lab. Thank you, Dr. Seke. Thank you, Sashi, Dr. Banerjee, Dr. Varia, Dr. Milanik, and also our committee from UNPAD. Prof. Tri and Pramdan have been supported me very well all these times. And it is because of their recommendation that I can go to Rutgers and study here and do some research together here. I would like to also thank my friends from Permias, the Indonesian Student Association in the United States, and also, last but not least, my lovely parents and brother. So that's all from me. Thank you so much.
Okay, what we'll do now is open this up to everybody except the thesis advisors for questions, and then we'll have the thesis committee examine Alvin a little bit more in detail. Okay, are there any questions from anyone except the thesis committee right now?
Thank you, Dr. Bertino. May I start?
Emily, do you have any comments or questions? You know this area.
Can I have my turn? Can I have my?
I think there's another students before me or another person, so I will wait my turn.
Okay.
Yeah.
Questions?
Questions?
Can I start?
Yes,
Dr. Bertino?
Yes, please.
Thank you for this opportunity. Firstly, I would like to appreciate, and we would like to express our gratitude to Dr. Bertino and all your staff for helping and supporting Alvin. Of course, we are proud that Alvin has done a lot, but maybe it's due to also your support. My question, Alvin, I think you have done a lot and you have presented all the experiments, but I'm very happy that you can prove that the synergy of NMPT and NMDK inhibitors are good synergic as anti-cancers. Of course, I'm happy that you have learned a lot of methodology and also the practical work, laboratory work. But the main questions, if you come back to Indonesia, you have to work very hard how to implement, how to boost our colleagues, our students, because our facilities are very restricted, very it's not enough, as you are lucky to have all your work at Rutgers. So my main question is how can you improve our capacity, our facility, based on your lucky experiment in Rutgers? Please, Alvin.
All right. It's a tough question from Prof. Panigoro. All right. Prof, I know that maybe we have some difficulties in establishing research in our institution in Indonesia, but I do believe that what you always teach me, as what you always taught me, that we can start it with a very simple research question, and we can start by try to inspire our students, try to learn, try to love research, and we can start with what we have in our faculty first, with all the fellowships that we have, before we try to search for any sophisticated modalities or try to learn about something that is not available in our institution. For something that is not available in our institution, that's the key of what Professor Ahmad always taught me about. It's important to have collaboration. So by establishing collaboration with other institutions and also collaborators, I think we can always boost the enthusiasm of our students and faculty to do research more and to produce delicate research, and that might be useful to help other people. Thank you.
Thank you very much, Alvin. We are looking forward for your your future development with us. Thank you, Alvin. Thank you, Brother Bertino.
Okay, thank you. Any other questions?
I have a question. Well, I wanted to first thank you for a great presentation that Alvin just did, and I wanted to thank him that he took the work Eugene started and make it better. So that's great. I have two questions to Alvin. Because I don't know the answers, maybe I should know the answers. So when you said that you have seen, as you increase the dosage of both of the drugs, you started to see additive effect rather than synergistic effect. I was wondering if the NAMPT and NADK inhibitor also use the same transporter, so that they are getting really start to compete with each other and not showing as much effect, whether you consider that. And I had another question. Do you want me to ask you both of the questions at the same time?
Do you think, Dr. Bertino, should I answer the first question first, or let Emily finish the question?
Okay.
Finish it. It's okay.
Okay, so my other one is that have you tried tryptophan depletion to reduce the amount of NAD or nicotinamide synthesis to have better synergy? That's another way of depleting the NAD from the cells. And it may be an easy way because it's an essential amino acid our body cannot make it. I was wondering if you consider that.
All right. Thank you, Amini. So yeah, for the first question, as I increase the additive effect of, as I increase the concentration and start to see additive effect of the treatment, well, yes, maybe you may be right. Maybe they share the same transporter, or maybe they share maybe they also share the same receptor of the cells, but I'm not sure because we didn't check for that transporter thing, but that might be a solution, an explanation why they start to show additive effect, that maybe they might be competing one another. And then about the tryptophan depletion, well, according to the literature that we searched before, the novel synthesis of NAD using tryptophan is only account for 1% of the NAD generated. So, that's why we didn't check for the tryptophan depletion in reducing the NAD, because like 1% might not be that significant like we inhibit the NAMT in the NAD sulfate pathway, because NAD sulfate pathway accounts for 90% of NAD production.
Okay, then maybe I don't remember well. I remember 40% of NAD is produced from tryptophan. So if it's only 1%, then I would agree with you. So sorry about this. Okay. Yeah. So that's okay. And my last it's
Sorry, can
it's
I
not
add
a
something? Add something? Okay, besides that 1%, if you take a look at the biosynthetic pathway picture, the diagram, we can also see that in the tryptophan is actually incorporated into the nicotinic acid pathway in order to generate NAD. So if we inhibit the nicotinic acid pathway, meaning that the dietary tryptophan will not be converted to the NAD
okay.
at the end. Yeah,
Okay,
that
makes a
might be
lot of
another
sense.
possible explanation for your question.
Yeah, and I think tryptophan is made actually maybe in the liver. And I mean not in the and NAD that is made from tryptophan is mainly in the liver. I should go back and check on that. This one it's not an answer or question. So I knew that isocitrate dehydrogenase can use NAD or NADP for its catalysis, but I did not know about alpha ketoglutarate dehydrogenase, and maybe I misunderstood that. Is NADP used in alpha ketoglutarate dehydrogenase as well as the isocitrate dehydrogenase?
So, so yeah, so according to the model that we develop and the model that we learn from, we can see that the question was NADP or NADPH?
Well, NADP becomes NADPH, right?
Okay, yes.
So whether alpha ketoglutarate dehydrogenase uses NADP as well as NAD as a substrate. I know that isocitrate dehydrogenase does. And I don't know. This is just curiosity.
So to
my
answer
part.
that, according to the paper that we cited in the review we published before, it stated that NADPH is used as the cofactor produced in the NAD in the alpha ketoglutarate reaction.
Your
Okay.
question
Uncommentable.
the substrate. Is the substrate NADP or is it NAD?
NADPH actually, the reduced NADP.
NADP.
Oh, it goes
Reduced
backward.
What's that, Emily?
It goes backward. That okay. I mean, this is not. It was just curiosity. And yeah. Thank you so much, Alvin. I really enjoy listening to you talk, and best of luck to you.
Thank you, Amini.
Are there any other questions?
Yes, Dr. Bertino, may I ask the question?
Okay. Please.
Yes. That I really appreciate that, Alvin, and it is a nice talk. And I have two questions. First question is, what is the best concentration that you recommend in your study for the NAD inhibitor? And this, because that I saw a lot of the drug cocktail, I mean the concentration that you use in this study, right? And the second question is that I saw that you did you did the sequencing. So did you find any domain have been mutated instead of the region that you point out in your protein alignment?
All right.
I think that's it. Yeah. Okay. Thank you.
Thank you, Bang Tora.
Yes.
So
and by the way, that's a great work. Okay. Okay. Thank you, Dr. Bertino.
Okay, go ahead.
So thank you, Dr. Pasaribu. Dr. Pasaribu is my roommate, actually.
Ah,
We share the apartment in New Brunswick. So the first question is the best presentation of the NAD inhibitor, right? Yeah, again, I would like to highlight the NAMT inhibitor with, I mean, yeah, the NAMT inhibitor as the NAMT inhibitor as the best drug here we use. And then about the sequencing, well, we actually focusing on that hotspot of R132H that has been reported to be the most frequent mutation happen in the IDH1 mutant cancers. So actually we only sequence at the time we only sequence the genes. We only sequence the gene that is actually responsible for that particular sequence of R132H position. So, but up to this time, I was surprised. I was also surprised to see that the C85 cell lines that we have since 20 years ago in the lab is mutant R132P mutant. It's a different kind of mutation that has been compared to the other mutation that has been reported in the Cancer Genome Atlas and another literature. Right.
Okay. Thank you, Alvin. Thank you, Dr. Bertino.
Okay. Any
May
other?
I? Yeah, may I, Dr. Bertino?
Yes.
Yeah, thank you. First of all, Alvin, congratulations. It's really nice work, lot of work. You did a lot in the lab. I can see from the result. So congratulations. So I have a few questions regarding the results. So first of all, I read briefly the review that you wrote. It's mentioned that this NAMT inhibitor somehow is failed in the clinical trial because of the adverse effect. So my question is, do you have any specific reason why you choose an ADP kinase inhibitor to combine with this NAMT inhibitor? Is it because the ADP kinase somehow inhibit specifically signaling pathway that can reduce the adverse effect, or is just merely you hope that the doses will be reduced by combining these two? So that's one. And the second one, if I see your Western blot of the cell line that you use, it looks like it's heterozygous mutation, right? Because you still have wild type allele and you have the mutant allele. So if your hypothesis is that this combination of drugs will effective in the mutant one, then why you didn't include cell line with homozygous wild type to prove your hypothesis? That's second. And third, you do the knockdown of the wild type IDH. So what I assume is by knockdown this wild type allele, you will have only the mutant. So I expect if this drug is really effective only for the mutant, the effect should be much more, let's say, effective in this knockdown cell. But I think it's the other way around, no? The result, or am I misunderstood? So maybe that's the three questions. Thank you.
All right. Thank you for the questions, Dr. Yunia. Okay. So this one about the NAMT inhibitor when it failed to the clinical trial, we learned that it might be because of the extracellular nicotinamide that might cause the extracellular nicotinamide that might cause the failure of the disease, as well as, and it's actually in line with the study, with studies that mentioning it's actually in line with a study that's saying that's stating that nicotinamide or NAD promotes cancer cell survival in the advanced stage of the disease. But by inhibiting NAMT, I hope that we hope that we can reduce the NAD generated. Well, NAD kinase, if we take a look at the profile of NAD kinase and the previous result that has been published before, we can see that NAD kinase also degraded. NAD kinase degrades the dihydrofolate reductase that is actually needed to stabilize the cells. So by combining both inhibitors, I do hope that we can decrease the NAD level and also degrades the dihydrofolate reductase, as well as reducing the NADPH generated from NAD. So we can have like a double reduce. I can say that it's a double decrease of NAD and NADPH reaction in the cells. By reducing both molecules, NAD and NADPH, I do hope that we can, we will have less NADPH to produce more oncometabolite, and we can also have less NAD and NADPH to promote cell proliferations, because according to our result, we also significantly decrease the ATP production in the cells. Again, and then your second question about the Western blot result, yes, it is. It looks like a heterozygous mutation because we have both cells are expressed, and we try to take a look at in the cancer genome atlas, and it's very difficult to find the homozygous cancers at the time. Yeah, and then your third questions about the wild type. Yes, so we knock down the IDH1 we knock down the wild type IDH1 using the shRNA, and then leaving the only mutant cells there. So your hypothesis was like the cells should be more sensitive, right?
Yes.
Yeah.
Yes.
But again,
If that's your hypothesis, I mean, if you said that this drug is more effective in the mutant one,
no, yeah, right.
Right, that's what your hypothesis is, right?
Yes, yeah. So by so my hypothesis for this experiment is actually if we knock down the wild type IDH1, then we will have the cells to be more resistant to the treatment, because less alpha-ketoglutarate will be developed. Less alpha-ketoglutarate means that there will be less D2-hydroxyglutarate. Less D2-hydroxyglutarate meaning less inhibition of the nicotinic acid pathway. Because
so meaning that the hypothesis of, so the hypothesis is not that the drug will effective in the mutant, but you need this heterozygous condition then, right? I mean, if the patient has homozygous mutation of this particular mutation, then it will not work then. So this is the conclusion?
Yes, you're right.
Okay,
thank you for pointing that out.
Top. Okay, thank you. Good job.
I want to add two things on the Alvin's answer for these questions. So first thing is that why we choose this combination for the NAMT and the NAD kinase, because our purpose was not to inhibit only the NAD, but NADPH. NADPH is used as a substrate for the mutant IDH1, the conversion of the alpha-ketoglutarate to ATP. So that was our purpose, to reduce the NADPH too. For the second question regarding the Western blot, since Alvin showed that the mutation is there, so our antibodies, particularly for that mutation, are 132H. And the other blot he showed, that is the actual IDH1. So that is not the wild type. We are not saying. We are saying that the IDH1 is there. So the second antibody is for the IDH1, and the other antibodies, particularly for the mutation part, are 132H. So we know that those are like we don't know whether they are heterozygous or whether they are homozygous, but since we were looking also in the literature that whether there is any other cell line in the colorectal cancer cell line, we chose the wild type IDH1. Recently we found that the HeLa cells is the IDH1 wild type without any mutation. So Alvin is also taking part for that cell line. So he's going to continue with that cell line also in order to make the paper as a. Yeah. And for the third question regarding the knockdown, we went to our CRISPR facility here in the CIMG. We needed to particularly knock out the IDH1 so that later we can reintroduce the wild type IDH1.
Yeah.
IDH1.
Yeah,
But-
that will be a nice idea. Yeah.
But since the CRISPR facility was busy and they said that it will take like a 1 year or like a 6 years.
Yeah.
It's not suitable for a master student.
Yeah.
For the master's.
You should stay longer there, Alvin.
Thank you, Gula.
Thank you.
That Alvin can handle himself, I'm sure. Any other questions?
Hey, maybe if I'm allowed to ask some question a little bit, Joy Maker here. I'm very interested with this cancer, and I hope this Alvin can bring back positively to Indonesia and also to spread it to the whole world, to the success of this NMPT. But yeah, focusing and this still to this line and NMPT and NAD. Alvin, what can you suggest that, or can you recommend based on your trials, based on your experiments, to the next scientist that would like to continue to your path and pursue on the details of this NPT? Maybe you have limitations on the time during your stay here in the US, but if you would like to suggest them, what can be done to improve or to achieve a better result, or even to minimize the side effect of this combination of this NPT and NAD to achieve a better result?
All right. Thank you, Danny.
Thank you.
Thank you, Danny, for the questions. If we're talking about the future recommendation for this result, and I do hope that we can continue further for, we have time. Actually, earlier this year, I think in January, I prepared a protocol for in vivo study, the animal model study. But however, due to COVID pandemic, we have to cancel that study, and I have to leave the Cancer Institute for about 4 months until they give me the permission to re-enter this building again. So again, yes, to continue the study and check for the vivo toxicity and efficacy will be very important before you go or proceed to the clinical trials. Yeah, that will be my recommendation for the nearest future. Thank you.
Okay, I think at this point we'll just ask the committee to stay around, and we're going to ask Alvin some more questions. Thank you all for attending his thesis defense, and I think we're really proud of Alvin, and we're going to grill him a little bit more. Thank you all. Thesis committee.
Everyone for coming.
Thank you.
Yeah. Would like to thank everyone coming from Indonesia and all my friends from the US. I would like to thank Dr. B. I came from the medical faculties, and I will from Universitas Pajajaran. I would like to thank, I think we have more than 10 professors from the Faculty of Medicine coming here at this Zoom. Yeah, I would like to thank Professor Parwati, Professor Kartasasmita, Professor Ahmadi, and then I would like also to thank Professor Maskun. I would like to thank a lot of Professor Agustiani from the psychology faculty also coming, and I would like to thank some of my high school teachers. They're coming here too. I would like to thank Ambassador Diantrian Sajani, and also the Mr. Consul, General Mr. Arifi Saiman. They're also the alumni from our university. They're working in the States now. I would like to thank Ambassador Juliana, and also Consul Simorangkir, and also all of my friends who made it here at this time. I would like to thank also Chief Hasan Sujono from Alumni America. Thank you, Chief, for coming. And I would like to thank everyone. So after this, I know that you will grill me more. So let's continue to the next session. And I think everyone is pleased to leave the Zoom conference.
Thank you all for coming, and we're really proud of Alvin, and I think he's going to be a great addition to your faculty in the near future. Thank you all for coming.
Thank you. Thank you. Bye, Alvin. Good luck.
The committee, please stay behind.
All the best, Alvin. Good luck.